Welcome to the Outsourced Pharma Capacity Update Partner Week, the first of 2025. I'm Perry Rarick, your host, and we're so glad to have you with us today. During this Partner Week, we have four different focus areas for each day of the week. Tomorrow is small molecule, Thursday is cell and gene therapy, and Friday is fill finish. However, today, our focus is large molecule drug substance development and manufacturing, and we have representatives from five outstanding contract development and manufacturing organizations. They're all with us today, and they're going to talk about their capabilities and capacity to help you advance your therapies. It's very exciting stuff. They are from Mabion, Cytovance Biologics, Catalent, Norway Biotech, and KBI Biopharma. Now, I invite you to just take a quick look at your event user interface. And there are a couple things I want to point out. One is in the center, kind of center right, there's a Q&A box. You can put your questions, just type in your questions and comments there. I encourage you to do that. I will see them, and the presenters will see them, and we'll try to address as many as possible. You can also contact the presenters directly. Down the left side of your screen, you'll see all the presenters and their contact information. You can reach out to them directly. In the upper right-hand corner, there's a box if you'd like to follow up with one of the CDMOs. You can just very easy click on that, and somebody will get back to you after the event. All right, if you have technical problems, perhaps your screen will freeze. Just hit refresh, and in most cases, you'll be right back into the event. And with all that, that's it. We are ready to go. Our first presenters are from Mabion. They are Nigel Stapleton. He is the Vice President of Business Development. Adita Bartuzik-Chubek. She is Division Manager for both analytics development as well as upstream and downstream process development. And Marta Bednarek, she is the Director of Manufacturing and Operations. Welcome to all of you.
Thank you very much. Let's see. Hi, I'm Nigel Stapleton. I'm Vice President of Business Development for Mabion, and I'm here to tell you a little bit about who we are and what we do. Mabion is a Polish CDMO. We're located in Łódź and neighboring Konstantinów, about an hour and a half away from Warsaw, the capital. Poland is a great place to do business. It's ideally located in the heart of Europe, and it's the sixth largest economy of Europe. We've been a member of the European Union since 2004 and a member of NATO since 1999. There are also direct flights to Warsaw from most major places, including New York. Poland has a very strong biopharma skill set. We have a well-educated talent pool coming from our 40+ higher education biotech-focused institutes, delivering the 5th highest number of graduates in the biopharma-related degrees in Europe. A little bit about us and our services. We have a history of 17 years, and in that time, we've built up a wealth of experience in development and manufacturing of biological recombinant proteins, but also in meeting pharmaceutical quality standards. We have undergone 11 GMP inspections, 8 GLP inspections. We are ISO certified, and we regularly get inspected by our clients and consultants for our GMP systems. Some words about our history. We were founded in 2007 and had our IPO on the Polish Stock Exchange in 2010. In 2015, we moved to our current facility in Łódź, and in 2020, we decided to have a strategic realignment and change the company from a company which, for the first 13 years of its existence, developed biosimilars all the way from scratch to commercial readiness, pivoting to a 100% pure play dedicated CDMO. So all the experience we developed in the first 13 years of our existence, we now offer to you, our valued clients. In 2021, Novavax trusted us with the commercial manufacture of their COVID vaccine. And it's a testament to our success that that relationship continues to today and is expanding. Meanwhile, we have added other clients to our services, including complex bispecific antibodies, hard-to-express proteins, and other modalities. Building upon success, we are always expanding our services, including in 2023, adding a new fill and finish line to our offering and an additional 2 2,000-liter bioreactors. Ooh. Of course, the most important thing in any company is our team. We have 213 full-time employees spread out over various departments, as you can see here. And we're very proud of our high employee retention, 98% in 2023. That means that we have a very stable team. And we have an ever-expanding base of knowledge which we can retain within the organization and which can serve you, our valued clients. Right, this is our main manufacturing facility. And let's see, I have a pointer here. Let's see if I can use that one properly. Here in the front is the QC area, with behind it, the process development and analytical development areas. The area here is the office space, with behind it, the main manufacturing area. Within the back here, the drug substance manufacturing area, and in the front, the drug product manufacturing area. On this side, we have the warehouse, where we can store both raw materials and consumables, but also final products. Joining me today are Marta, our head of manufacturing. She will be joining me from the manufacturing area, and Edita, our head of process development and R&D, who will be joining me from the process development area. Welcome, Edita. I see that you're in place, so over to you.
Thank you, Nigel. Hello, everyone. My name is Edita, and I would like to tell you today a few words about our development capabilities. As we all know, the development pathway for biological drugs starts with high-producing cell line. Based on this cell line, a manufacturing process is developed and then transferred to large-scale GMP manufacturing in order to supply quantities of the protein necessary for next stages of project development. Our upstream and downstream development services are designed to create repeatable and easy scalable process that ensures that your product is manufactured in consistent and repeatable way. We have knowledge and experience in developing USP and DSP processes for biosimilars, where we have to obtain exact design quality of the protein, but also for innovative drugs, where we define the physicochemical characteristics of the protein that ensures the activity of the molecule. In this regard, multispecifics are very interesting because we have to ensure that the protein will be active against several different antigens. In our facility, we have capabilities to develop USP and DSP processes in milliliter scale and scale them up to liters in laboratory and manufacturing scale. Thanks to our knowledge, equipment, and also software dedicated to our solutions, we are able to transfer and scale up the processes in few runs. Thanks to having easy scalable equipment, we are also able to conduct in effective way process characteristic studies. Thank you, Nigel, and I'm passing voice back to you.
Super, thank you very much, Edita. Marta, are you ready in the manufacturing area?
Thanks, Nigel. Yes, I am. Hello, everyone. Last time we've met in upstream area, bioreactor room. Can't be there right now due to ongoing work, and we are working on a great innovative project that can actually make a difference in oncological treatment. In fact, for this project, we have introduced some new equipment within downstream part, like an ACTA pilot, for an instance. Coming back to the point, as we last explained, we were focusing on large-scale production, range from 1 to 2,000 liters with capacity of 2,000-liter bioreactors. Now, as a part of the current project, we have modified our configuration a little bit adding the capacity to manufacture smaller scale GMP batches from about 100 liters upwards. This is also an interesting future for perfusion technology, and our bioreactors are perfusion ready. In parallel, we are developing our DP capabilities, where we have added a new vial format for filling application to our service offering. Thank you.
Thank you very much, Marta. Now, Edita has now moved to a different area, so I'm wondering whether Edita is already in place or is still running through our facility. Edita, are you in place?
Yes, I'm in place.
Very good.
Yes,
I hope you're not too out of breath from running.
Should be good. So now I'm in the analytical area. At Mabion, we possess a wide portfolio of analytical methods that allows us to conduct in-process control, as well as the SDP quality testing and reference drug product characteristics. Our USP, DSP, and analytical platforms together form a comprehensive manufacturing platform that can be utilized by different biotech companies, regardless of their size, product characteristics, or development stage. Thanks to our wide range of analytical portfolio and also our problem-solving attitude, we are able to conduct seamless analytical methods transfer, use available methods with or without adjustments, or develop product-specific analytics de novo. It is worth to mention that in the past, our analytical platform was very much focused on biosimilars, but recently we have been working with very interesting, innovative bispecific molecule, and we also adapted our analytical platform to deal with that. Thank you.
Thank you both, Edita and Marta. Back to me. Right, as you have now seen two of our experts and seen some of our facility, please allow me to tell you something about the services that we offer. Mabion offers a full end-to-end service. We're active in process development. We offer quite uniquely preclinical and clinical analytics, and we can offer clinical and commercial manufacture of both drug substance and drug product. Supporting these activities or offered as part of a standalone service, we can offer our extensive experience in characterization and our regulatory expertise to our clients. In the upstream arena, in the drug substance manufacturing upstream arena, we're experienced working with both mammalian and insect cells. And our main production bioreactors are 2 × 2,000 liters Cytiva bioreactors or 2 × 2,500 liters Kuna shaker orbital shaken bioreactors, each with their own dedicated seed train. Our downstream, our bulk harvest technology is either based on pod filtration or centrifugation, depending on what the process needs. For downstream purification, we have a wide range of technologies in a very flexible setup. As Marta explained, we can go from very small to very large batch sizes. Importantly, in our manufacturing facility, we have quite extensive capabilities for buffer preparation and media preparation. This makes us less dependent on the timelines of suppliers. Let's see, next slide. As you saw during Martha's presentation and you heard in the background, we offer state-of-the-art fill and finish from our brand new filling line, which together with our drug substance creates a robust end-to-end service offering. Our filling line can handle all the common formats from 2R to 100 milliliters, and it's equipped with isolator technology. As we offer both drug substance and drug product under one roof, this substantially reduces the risk and increases the robustness of our services. And as Edita so eloquently explained, our process development department can develop both upstream and downstream processes. And by combining process development, analytical development with a statistical design of experiment approach, we can efficiently develop high yield, high quality processes. Alternatively, if you have an in-house developed process, we can transfer it in, optimize it if necessary, scale it up, and use that as a basis for first your clinical and then your commercial manufacturing. Our analytical method development department, which you also saw at the background of Edita's presentation, uses our broad and comprehensive analytical platform as a starting point to develop a full suite of analytical and QC methods specific to your particular molecule. We can qualify those methods for clinical manufacturing or fully validate them for commercial support. These services, including reference standard characterization, comparability, and similarity studies, are offered either as a part of a larger project or, if you need them, as standalone services. Finally, designed to fit seamlessly together, our services offer an end-to-end solution for clinical manufacturing, starting with cell banking through process development, analytical development, drug substance manufacturing, fill and finish, QC release, stability studies, and anything else you might need for your CMC needs of your clinical projects. We can help you get through that. And as you move towards commercial manufacturing, it's reassuring to you, our valued clients, that you can rely on our broad experience in process characterization and PPQ studies, as well as commercial supply, to continue the relationship we built over the years. The experience that we've built over the last 17 years, first developing and manufacturing our own products, but now serving our clients, all the way from an idea to commercially ready products, is now at your disposal to help you successfully navigate your way to the market. And that was all we had to say from the presentation, and we are very curious as to what questions you might have.
Thank you, Nigel. Great presentation. So a few questions. So personalized medicines, when they commercialize, aren't necessarily in need of large volume production. And you had described some of your investment into smaller scale production. Can you tech transfer those type of things on? What does that look like? How quickly can you get those projects started?
Edita, is that a question that you can pick up? Let's see if Edita's still here.
Yes, I'm here, but I think that this is a question more dedicated to you, Nigel.
Right, okay, I'll pick it up. Yes, as Marta has explained, we can offer both small scale and large scale. And for personalized medicine, where smaller batches are typically the norm, we have a 50-liter bioreactor and a downstream process, which is very, very flexible. So we can bring that in-house via Edita's process development department, optimize it, make it GMP ready. Typically, these processes, when developed in more research-oriented companies, need a little bit of fine-tuning in order to make them ready for GMP manufacturing, and then develop at the scale that you need. Typically, this can be quite a fast, efficient process, but it very much depends on how ready the process is from the client that we can get, and how much analytics needs to be developed to support it. Okay.
Also, do you have the ability to, if somebody has the need for development and production of complex recombinant proteins, do you have the ability to support that?
Absolutely, that's the core of what we do. We work with partners for the cell line development part, but after that, we are exactly geared out for the development of processes, analytics, scale up and manufacturing of recombinant proteins, including drug product manufacturing, release, stability. So basically really end-to-end CMC service for exactly this purpose.
Excellent. Okay, well, thank you very much. Great presentation. Thank you for hanging with us late in your day there in Europe. So great job. Mabion, Nigel Stapleton, Edita Bartuszek-Czupek, and Marta Bednarek. Great to have you with us.
Thank you very much. Have a great day.
Quite a number of you joined us after we started, and so I just want to Make sure you understand that there's a Q&A box in the center of your screen, center right. Please type in your questions and comments. I will see them. The presenters will see them, and we'll address as many as possible. Down the left-hand side, there's contact information. You can reach out directly to the presenters. So we encourage your participation in this event.